Hello, everybody, and welcome to the 35th Great Lakes Phragmites Collaborative webinar. And this webinar is part of our Applied Techniques webinar series. Our presenters today are Laura Borgeau-Chavez in collaboration with Phyllis Higman and Amanda Grimm. I'm Elaine Ferrier from the Great Lakes Commission, and I'm hosting the webinar today on behalf of the Great Lakes Phragmites Collaborative. The collaborative is a partnership to link people, information, and action on invasive Phragmites. We are coordinated jointly by the Great Lakes Commission and USGS Great Lakes Science Center through funding from the Great Lakes Restoration Initiative. We aim to facilitate communication among stakeholders across the region, assist in the development of needed tools and programs, and serve as a resource center for information on Phragmites biology, management, and research. We are led by a diverse advisory committee and are organized according to the principles of collective impact. Check out the resources on our website, including management guidance and past webinars. Join our listserv to connect with hundreds of other Phragmites professionals, and sign up for our weekly newsletter, which includes a monthly research roundup of academic papers, as well as a weekly newsletter and roundup of social media activity. Today's webinar is being recorded, and it will be uploaded to our YouTube channel, as well as our website. You can access an archive of past presentations on our website at greatlakesphragmites.net, and you can also view and register for upcoming webinar presentations. Now it's my pleasure to introduce our presenters today. Phyllis Higman, who will be kicking us off, is a botanist and senior conservation scientist at the Michigan Natural Features Inventory. And Phyllis just celebrated a 25-year work anniversary, so congrats to Phyllis. And Dr. Laura Borgeau-Chavez, I apologize, there's a typo on the slide here. Laura is a senior research scientist and adjunct assistant professor of Michigan Technological University. And Amanda Grimm is a research scientist also with the Michigan Technological University. And with that, I will transfer over control to you, Phyllis. Just a moment here. Get started, just transferring. Okay. All right, and if you can just share your screen, perfect, it's coming through great. All right, thank you, Phyllis.
So you can see everything now? Can you hear me?
Yes, you sound great, and we can see the slides.
Great, awesome. Good morning, everybody. I'm gonna be doing a little brief overview of our monitoring protocols, and then we'll be having Laura Borgeau-Chavez talking more. Amanda will be going next to summarize some of the things that I've talked about, and then we'll be passing the baton to Laura for the last part of the talk. You can see the agenda there. We're going to talk a little bit about the motivation for the project, a brief overview of our goals, describe the field monitoring protocols that we tested, and then compare them. And then we'll move into the remote sensing protocols, including worldview imagery and some drone work. And then finally, we will wrap up and discussion, have questions. And I think we all realize that we need to keep this conversation going and connect across the country to learn more from one another. It's actually part of an overarching effort that involved two projects that Laura is the principal investigator on: implementing adaptive management and monitoring for restoration of invasive Phragmites, which was an EPA funded grant. And then we had a second grant that basically enabled us to do a larger area and add additional activities that was funded by Michigan Invasive Species Grant Program. And then at the same time, we had another project that was ongoing there by Laura Ogar of Bay County, which was doing some large scale treatment. So we hooked all these three together, and we ended up treating over 338 acres, which were treated and monitored for 2 or more years. And we also were able to provide cost share for another 235 acres or so. We had a large project team: Laura from Michigan Tech, myself from Michigan Natural Features Inventory and Michigan State University, Ken from Northern Iowa, Bill Curry from the University of Michigan, Jason Martina from Texas, and then the collaborators included the U.S. Fish and Wildlife Service, Bay County, and many folks from the Saginaw Bay SISMA that really did the brunt of the on-the-ground work. So why did we start this project? There have been several key articles that have critiqued our current managements across the country for Phragmites, basically stating that they're often focused on single or groups of Phragmites patches rather than a larger site that it's contained in. They often lack explicit management goals. Often they lack multi-year planning. High-resolution Phragmites maps are improving, but have been often not used in projects adequately. And then there is frequently no monitoring or inadequate monitoring, and often results are expressed as sort of a goal level, oh yes, we think they succeeded or not. So this is improving, as we've all read the criticism, but I think that down the last 3 points are things that we still really need to focus on. We rarely measure the ecosystem response. A lot of projects are just looking at the kill of Phragmites. They're very rarely considering the entire landscape, and they rarely address the root causes of invasion, such as nutrient inputs into the systems that have been invaded. With all of these things, we're spending millions of dollars controlling Phragmites infested wetlands every year, and we need to be able to demonstrate that these funds are achieving the right results. So, although we've had these criticisms, it doesn't mean we aren't achieving success. Anybody who's tried to work with Phragmites has probably felt like this individual rolling the stone up the hill. It's challenging. We certainly do have success stories, but we may be wasting money in some places by using ineffective or less than optimal treatments and sequences of treatments. There may be unintended consequences that could be avoided. And very importantly, we can't tell our true story to critics and funders. And in order to continue this work, we need to be able to demonstrate through monitoring our success. And then finally, with all of this effort going on to treat Phragmites, by not having solid monitoring programs in there, we're not taking full advantage of learning from what we do, which is critical to improving success. So we're looking for ways to get monitoring done to better tell our story. And as I said, we certainly do have success. Here's a couple of examples for this project. At the top, you see the before picture, and at the bottom, you see the after picture. So there's been very significant results in this short time period. Questions need to be answered, though. Was this the best site to be doing this treatment? What's coming back in? Yeah, we got a good frag kill, but did we really succeed in what our management goal was? We also have some areas where there's been unanticipated consequences. Here's a frag stand we treated with a traditional herbicide and mowing, I guess it was, or excuse me, crushing. And this is what it looked like after that was done. And for those of you who aren't familiar, this is the European froglet that has recently been showing up in the Great Lakes. And I'm not sure which is better, the stand of frags that was there first or the European froglet that came in after. So the most important thing is that we learn from this work so we can respond to these new changes effectively. Our project goals for the overarching group of project was to build a partnership amongst lots of different people and to develop a regional long-term strategic plan for the Bay instead of doing it on a site-by-site basis. And the third goal I have down there is my own personal goal, which is to conserve the biodiversity of Saginaw Bay. This is home to some of our last remaining lake plain prairies, and I hope that we can maintain them. I encourage you to take a look at the larger report because we won't be talking about many of the things we did here. There's a lot of information that could probably be useful for managers on the ground. Just to give you a broad picture of what we're looking at, we did broad scale mapping of the land covers and high resolution pre-treatment mapping, which allow us to look at outliers, sources, and pathways. We conducted landscape and connectivity modeling. We had meetings with stakeholders. We fed this information into an ecosystem model, boundary and wetland modeling, and then we're able to look at different scenarios to see which would result in the best success. The managers made their treatments for their sites, the crews were hired, and then we had the evaluative component here, which of course then feeds back into the original work. Just a quick note, the landscape modeling included looking at nitrogen loading, water levels, and field pressure and how they move across the landscape. This then was informed the Mondrian modeling, which looked at multiple complicated factors in it. And the ultimate goal, of course, was to look at site to provide site-specific management of best treatments and sequences based on the information gathered here. So we have the feedback loop where we do all the work, we look at what happened, we go back and redo it, and then this gets repeated and we improve them from what we learn as we go along. This project is not talking about this larger picture. It's another 2 or 3 webinars to get into the details of these. We're talking specifically about the monitoring that we tested and comparing them. Our project was located in Saginaw Bay in central Michigan on the east side. The yellow here is the known occurrences of Phragmites. The orange were sites that were treated for the sister project, and the green are the sites that were for the our 2 projects. So our monitoring goal was to find protocols that are practical enough that they will be used, but that they also will provide data we need to tell our stories of success or failure. Phragmites is not easy to manage, but it is essential that we do it so we can improve how we're managing it. We reviewed the Pier literature, Gray literature. We had a stakeholder meeting to gather input from practitioners on the ground. We tested several protocols of varying intensity, and then we compared them. An overview is Tier 1. We tried to do simple to complicated. Tier 1 is a qualitative photo monitoring method. And then Tier 2, which was the most challenging, and what we were really after was where it doesn't require super expertise in plants, but gets you the data you need. So we did a quantitative belt transect, and then we modified that and added some plots across the transects. And then finally, the Tier 3, we used the Great Lakes Coastal Wetland Monitoring Protocol. And I'm going to go into each of those now in a little more detail here. Because we noted that we often are criticized for not having management goals, we did set this management goal here. Our early detection sites success was totally eradicated from the Phragmites in isolated patches that were less than 1/4 acre in size. And then for the larger stands of Phragmites, success was a 60% reduction in Phragmites in year 1. And then stability after there's no herbicide required for 3 years. And then our desired condition is that we have native species returning. So our Tier 1 monitoring, probably most of you are familiar with this, the photo monitoring. It's relatively simple and easy to implement. It's a qualitative protocol. Encourage you to put your management goal right up there in the top of the form. We gather data about the site, who's sampling, and so forth. And then we have the sampler estimate the percent cover of invasive Phragmites and desirable species and undesirable species. And these are qualitative estimates. These can be customized by the site and management goals. Our thought here is it's easier to learn a few distinct invasive species than it is to learn all native species. So this protocol can actually be implemented by people with relatively little experience. The photo points are permanent for repeatability. We take 3 pictures, one through the center of the patch and one to either side. And of course, you may take additional photos as desired for certain areas of your site. And you need to use the same photos and cameras, etc., year after year. We also record the details of the treatment and monitoring. We identify the natural community type, and that's dependent upon your management goal. You may change what this measure is here. And of course, access notes are helpful for those who do monitoring in the future. We also included a second part to this where we measured aesthetic impact, recreational impact, and safety impact to make this protocol a little more informative, and we borrowed the ranking definitions from the new DEQ Eagle, Phragmites Treatment Management Prioritization Tool. So each of the impacts, aesthetic, recreational, and safety impact, have definitions for severe, moderate, and mild. And the example here for aesthetics is below. So you simply look at the different categories and score your site based on those. So tier 1 is the bare minimum for every treatment, and it needs to be assessed over time to be useful, but eventually you have to actually get into the Phragmites stand. It's very clear from our work, and I'm sure others, if you stand at the edge of a Phragmites stand like this, it looks like there's 100% Phragmites, but if you walk the transect through it all, there typically will be a lot of variation. Thank goodness we had Emily Schaefer to help us go through these stands and keep our spirits up. It's not easy work. So moving on to tier 2 monitoring, this is what I was most excited about, trying to get protocols that go beyond a simple photo monitoring, but are able to be conducted by people who don't have a super expertise in plant identification. So we gathered this from literature. Various different folks have tried these belt transects. The sampler walks from the entire transect, recording the wetland zone and dominant cover every meter. Their assistant behind them then records that on a sheet of paper. The zones are the 3 major zones of a typical wetland. They may not always be present. And then we recorded along the transect whether it was dominated by live Phragmites, dead Phragmites, desirable species or undesirable species, open water, and bare soil. So they just marked these numbers down as they moved along, and this thing gives you a relative frequency of each of those factors. Examples of undesirables include cattail, flowering rust, or European frogbit, and desirables, native species. And these can be customized to the site and the specific management goals, as well as the skill of the sampler. For our project, we used all of the watch list species for Michigan, Great Lakes, and as our undesirable species. We used native species as those in the desirable category. You can, of course, specify these according to your desired future condition. And again, the whole point of this is, are we achieving our management goals? And secondly, is there a secondary invader lurking nearby? This is turning out to be one of the biggest challenges in Phragmites management, is other native or non-native species coming in after the Phragmites kill. Monitoring long transects through thick Phragmites gets old quickly, especially in July and August. It's hard to breathe, it's easy to panic, and it's easy to get lost. And as we thought about it, the belt transects gives you one measure, but since we're going through these stands, let's get a little more data while we're out there, because we certainly are not going back through that again. So we added a second part. That should be tier 2B up there, I'm sorry. We added 5 evenly spaced plots throughout the transects that the belt transects were being accomplished. And we divided the 1 meter quadrat into 4 different sections. And in those 4 sections, we measured the basal diameter of the largest stem in each plot, as well as the height estimated for those same stems. We also did a stem count. And these pieces of information here give us information about the biomass of Phragmites. We also did a percent cover of Phragmites in the entire plot and a percent cover of morphotypes. So this gives you some diversity measure, but it's able to be done by a person who doesn't have super high experience with species identification. We also took soil samples and leaves from the stems to measure nitrogen, which was part of our larger project. There's evidence that high nitrogen levels actually promote invasion and limit, if beyond a certain threshold, the ability to control it. So a summary of the 2B monitoring, we added 5 plot zone quadrat data. We took standard measures of these items here. This provides a relative cover of the Phragmites, dead Phragmites, desirable species, undesirable species, open water, and bare ground. And this provides more than just a simple estimate. It actually is measuring the desirable species as well as Phragmites kill. And most importantly, it forces the crew to walk through an entire Phragmites stand and capture the variation that you cannot see from the edge in many cases. Be sure to learn how to figure your points between samples before you go out there. It's very difficult once you're out there to think straight while you're walking through these dense stands. And here we are going through post-treatment. And you can see on the right, we actually have 2 crews going at the same time, and they lapped over one another as they moved through the stand. This was not an easy task in many sites. This is very difficult to walk on, easy to fall through, and I think sometimes not even safe. So the team rebelled in our large sites, and we drew a line at 500-foot transects and determined that when the sites were that large, we would use aerial imagery and drones rather than field walking through the site. Our last monitoring tier is 3, and again, we use the Great Lakes Coastal Wetland Monitoring Program protocols, which have 3 transects perpendicular to the water gradient from wet meadow to emergent and then submergent marsh. Within each of those wetland zones, there were 5 1-meter plats evenly spaced for a total of 15 plots. If the zones were too narrow, the transect was placed perpendicular to the original transect across the middle of the zone for 15 meters in each direction, and plots were sampled at 7, 12, 17, 24, and 27 meter. Abiotics factors and percent cover of all species were measured in this case. So here's where you have the highest level of skill for plant ID that's required. A rake was passed across the bottom of the quadrat to capture species in the substrate, and all unknowns are collected and systematically labeled for later identification. This is an example of what the sample form looks like, and you simply put your notes in each little box to the corresponding question. And the species list is obviously a lot longer than these 4 species. I wanted to mention one other protocol that we didn't have time to assess. This is the point line intercept method. This has been utilized very effectively by DNR parks at Bay City State Park. You have a series of transect across the water gradient, and you run a measuring tape along the transect, and at specified intervals you identify those plants that intercept the line. And this will enable you to get a relative frequency of all species to measure change over time. And this is a relatively quick method, but it also requires high knowledge of plant ID. So Amanda will be now taking these different tiers of monitoring and comparing them. I just wanted to thank the Great Lakes Phragmites Collaborative for all the information and resources that are provided, and all of those who contribute to that. And I want to thank Pam Grassmick from Beaver Island for her inspiration. She mounted a response to Phragmites on Beaver Island, and they now are a complete maintenance mode with little Phragmites at all. Ironically, she's standing on Trout Island within a huge infestation of spotted knapweed. And again, I want to remind us all why we do this. There's a reason for it. We need all the pieces that we have out there because that provides greater resiliency to our ecosystems. And last but not least, I encourage you to get out, take a look at what's blooming right now, and perhaps you'll be able to take in some of the spring flora. So with that, I'll hand that over then to, or I'll ask Elaine then to move on to Amanda.
Thanks, Phyllis. I'll just transfer control over to you, Amanda, now.
Thanks.
Should see the pop-up in the moment there. Looks great.
Okay, great. So just to review what Phyllis mentioned earlier, we had kind of 2 grant programs that allowed us to treat sites in Saginaw Bay, both of which resulted in 4 sites that were treated in 2016. The first group was what we'll call the EPA sites, funded by EPA GLRI grant funding, and those were treated with a combination of glyphosate and Signet Plus, applied with a boom sprayer in the fall of 2016, and then all of those sites were mowed before the 2017 growing season, followed by spot treatment in the fall of 2017, and then one site was subjected to some mulching at 2017. Our other group of sites, we're calling the MDNR sites, were funded through a Michigan Invasive Species Grant Program project, and those were sprayed with a mix of glyphosate and imazapyr in September 2016. And the intent was to also mow those before the 2017 growing season, but that didn't end up being possible. So 2 of the sites were partly mowed before the 2018 growing season. So originally, our planned difference between the 2 groups of sites was glyphosate plus surfactant versus glyphosate plus imazapyr, in part related to specific concerns identified from previous management efforts that the use of imazapyr could negatively affect the native seed bank, and concern that glyphosate alone would not be sufficient to achieve the desired Phragmites kill. What we ended up with, due to site conditions, was 2 differences between these groups of sites, imazapyr versus no imazapyr, and biomass removal via mowing versus no mowing at the MDNR sites. Taking kind of a closer look at what some of these sites look like, these are the satellite-based classifications of dominant cover at the MDNR sites before they were treated. So towards the south end of Saginaw Bay, we have the Hampton and Vanderbilt sites, which were sort of the worst case scenario, almost a kilometer-wide band of solid Phragmites. On this map, Phragmites is that purple color. And then we also had some smaller sites up around the Pine River and the Saguenay River on the west side of Saginaw Bay, and those also had some significant components of Phragmites, but in more of a matrix of other cover types as well. Our EPA GLRI treatment sites were clustered more down in Bay City at the south side of Saginaw Bay. We had Callahan, which was more similar to Hampton and Vanderbilt in that there was a lot of Phragmites kind of right along the coast. And then we have these more inland sites at Putz Park and Dutch Creek. And on the right, you can see the pretreatment land cover at Dutch Creek. So again, that dark purple is big patches of Phragmites. So a lot of pretreatment Phragmites cover at these sites as well. And across the 2 groups, we had pretty similar percent cover of Phragmites. So here's an example of just photo monitoring before versus 1 year after at the Ted Putz Park site of the EPA group. Before we get into results, I wanted to talk a little bit more about the UAV-based monitoring we did. Phyllis was discussing the safety concerns that came up at some of these sites. some of the transects that we had originally planned to apply Tier 3 monitoring, we eventually had to change that plan because it was just too much of a safety concern. If someone became injured or heat stressed a kilometer out into the Phragmites, it would be very difficult to get them out if they couldn't do it under their own power. So we replaced some of the Tier 3 monitoring with flying a small UAV over the transects and classifying that imagery. So here's a comparison of pre-treatment and post-treatment imagery of one of those transects. On the right, the 2017 imagery, we kind of captured the edge between where the site was sprayed and the treatment boundary, where you can still see the very happy Phragmites. So the difference between treated and not treated, very obvious. When we flew these sites, we collected, field notes and photos to document what the dominant vegetation types were, and then used that information to classify the mosaicked transects of UAV imagery using an object-based classification method. Here's an example of what that classification looks like with a few comparisons to what the field photos were for those sites. This is one of our Hampton transects where the transects were about a kilometer long and mostly very dense Phragmites before treatment. Here's an example time series of kind of what these UAV classified imagery transects looked like over time. On the left, we have pre-treatment, almost solid, dense Phragmites. 2017, after spraying, but before Hampton had been mowed, we have a lot of detritus, so pretty much standing dead Phragmites, with you can see a few patches of remnant live frag, especially out at the water's edge where they didn't quite catch it. And then on the right in 2018, a lot of the site has been mowed, or the dead Phragmites has fallen over and it's converted to more of an aquatic bed dominated area. So starting into the results, because our definition of treatment success focused primarily on decrease in percent cover of Phragmites, we were interested in how these protocols compared in terms of the estimated cover of Phragmites. So we compared all of them to the site-wide percent cover of Phragmites derived from the satellite remote sensing map, which Laura will discuss a little bit later. That satellite map isn't, 100% infallible, but it's been very well validated, so we use that as our quote-unquote ground truth. T1 is the kind of eyeballing Tier 1 percent cover from one location, so not walking into the site. And as you might expect, there was a much larger difference between Tier 1 and the site-wide satellite map. We had smaller differences between the Tier 2, where you actually walk a belt transect through the site, and the site-wide map. And then we still ended up with a fairly substantial difference between UAS and satellite maps, even though they're both based on high-resolution imagery. Because UAS, we were just mapping along the transect. So that's reflective of the difference between just looking at a few transects versus looking at the site as a whole. One thing we were interested in was whether Tier 1 kind of eyeballing the percent cover might be less variable for small sites. You would think intuitively that for a small site you could get a better, just visual estimate than for a very large site where you're trying to estimate percent cover from one location. But from the graph on the right, we didn't really see that. The Tier 1 eyeball estimates were pretty variable, both for very small sites and very large sites. It's just difficult to come up with an accurate estimate of, 12-foot high vegetation from the ground. So overall, we just found that going through the site is going to give you a much better estimate of how much your Phragmites cover has actually changed compared to eyeball, even for small sites. So that's reflected in our estimates of how much Phragmites cover decreased 2 years after treatment. going back to our definition of success, we were looking for at least a 60% decrease in Phragmites cover. And the two right plots, based on the Tier 2 belt transects and the Tier 2 plots, they indicate that across all of our sites, both EPA without imazapyr and DNR with imazapyr, we achieved at least that 60%. The rightmost plot, where we're just looking at the 5 plots along the belt transects, it looks like we had lower success for EPA just because we did have some regrowth in a couple of the plots at one site at Callahan. And because we only have 5 plots, that, kind of biases us in the direction of less Phragmites kill. So that's an example of how the belt transect and the 5 plots along the belt transect work really well together, because the belt transect gives you a much better idea of kind of the site-wide conditions, whereas the plots, while they can give you really useful information about density and kind of percent cover of different types and what's regrowing, they can give you a biased estimate of frag kill if you were using them on their own. So it's a good combination. And then on the left, if we were just doing the eyeball, we would conclude that at one of our sites, Pine River, we didn't reach our definition of success because our eyeball-based estimate indicated a Phragmites reduction of only 55%, but all of our other protocols are indicating the opposite. So an indicator that we need a little bit more than just the eyeball cover estimate. As Phyllis described, our Tier 1 photo monitoring and eyeball estimates protocol also included estimates of human values impacts. So for all 3 of these plots, for the aesthetic, recreational, and safety impacts, blue is the EPA sites and orange is the MDNR sites. You can see that for all 3 impact categories, we had large decreases in the average impact, where 3 is the severe impact and 0 is no impact, from pre-treatment to 1 year post-treatment, and then continuing to decrease from 1 year to 2 years post-treatment. And then for the orange MDNR bars, we have much less change, and that's really reflective of the difference between mowing the sites shortly after treatment versus not mowing, because just the standing biomass poses such a significant effect on the aesthetics and safety of the site. Tier 2 lets us get a much better sense than Tier 1 of what is growing in place of the treated Phragmites. Both the belt transects and the 5 plots along the belt transect indicated a kind of consistent increase in the percentage of the sites dominated by desirable vegetation for the EPA sites. For the MDNR sites, we have kind of more of a mixed pattern from pre-treatment to post-treatment and then year 2 post-treatment. And again, because we have this kind of combination of differences in herbicide combination and biomass removal between sites, it's difficult to say 100% for sure whether this is more an effect of the delayed and partial biomass removal at the MDNR sites, or whether the presence or absence of imazapyr in the herbicide combination could be having an effect here too. Another thing that we can do with the Tier 2 data that we found really useful is, because the 5 Tier 2 plots include stem counts of Phragmites in the 1 square meter plot and measurements of height and diameter, it allows us to use an allometric equation that relates height and diameter to biomass to estimate changes in live Phragmites above ground biomass. Before treatment, Phragmites at the EPA sites averaged slightly more than 2 kilograms of dry weight per square meter and 1.5 kilograms of dry weight per square meter at the MDNR sites, and then both 1 and 2 years after treatment. And you can, the bars are barely there. Live Phragmites biomass was negligible at both sets of sites, as any of the live Phragmites that remained was small and occurred at a much lower density than pre-treatment. Finally, we did the Great Lakes Wetland Protocol style Tier 3 surveys only at 3 of the sites, all of which were in the MDNR group of sites that included a Pier. We saw ranging from a small to a larger decrease in both total and native species richness across all 3 sites between pre-treatment and 1 year post-treatment, with kind of small variable changes from year 1 to year 2. So really we would need to do kind of some longer term monitoring to see what the actual effect on species richness is over time. Because we gather such detailed species identification information for Tier 3, we can also derive some more advanced conservation metrics from that data, such as the mean conservatism index, where species are assigned conservatism coefficients of 0 to 10, and the mean C values of all of the species present at the site are averaged to obtain an overall value. So higher mean C values for a site indicate a site with more conservation value, and then cover weighted mean C is similar, but accounting for the percent cover of these different species. Since we only had 3 sites, we kind of see different patterns across the 3 sites, even though they were subjected to the same treatment. So we don't see much change at Hampton Township. We see a significant increase in mean C at the Saginaw River mouth site, and then just variation at the Vanderbilt site. So to kind of get some stronger conclusions from this type of monitoring, it'd be really useful to have more sites to look at and over a longer period of time again. And then finally, floristic quality index is another value that we can look at that is maybe the most appropriate metric for these kinds of sites, because there's variation along Saginaw Bay in terms of the site history and nutrient inputs and that kind of thing. And that uses the mean conservatism index and incorporates the total species richness. And again, we kind of see the same difference across the 3 sites with not much change at Hampton, increasing at Saginaw River, and then varying values at Vanderbilt. So to summarize, we compared these protocols, the Tier 1 protocol of photo monitoring and kind of the eyeball dominant cover. It's easy to do. There's low effort, skill, safety hazard involved. It does give you this additional information on the human impacts of Phragmites, which is kind of a useful extra bit of information that you might add to a different monitoring protocol if you found that useful. And it gives you an estimate of Phragmites cover, although, as we can see, it isn't necessarily a highly accurate estimate. We found the combined Tier 2 protocol, where you do the belt transect and the plots, to be really useful. It was kind of an intermediate level of effort and skill. It does pose somewhat more of a safety hazard because you do have to walk through the site. But it gives you a better estimate of Phragmites cover that's more comparable to a site-wide determination, and gives you some information about the change in the total undesirable vegetation, including other non-native species. You get some information about what's regrowing in place of the Phragmites, and you can approximate species diversity based on the morphospecies information. And then you can also estimate frag biomass, which can be useful to compare 2% cover if you have large areas where there is still Phragmites, but it is much smaller and less dominant. That's useful to be able to look at biomass rather than cover. Tier 3 gives you the most information if your management goals focus on biodiversity or restoration of a high-value site. That can be critical information to have, but it also requires a trained botanist and a lot more inputs in terms of the time required to do that monitoring and the skill involved. And finally, UAVs give you a good estimate of the dominant cover types. It's fairly fast to do, but there is a high kind of startup cost of access to the UAV and a pilot and someone who can classify that imagery. A little bit more detail on our conclusions about using UAV imagery for biodiversity. Obviously, it's easier to identify taller or overstory vegetation from aerial imagery. It's possible that if you're only using UAVs and are traversing the site, there could be secondary invaders such as Phragmites that you might miss because they're in an understory and aren't visible from above. You can differentiate dominant vegetation types, but generally not to species level. So you can't necessarily distinguish between broadleaf and narrowleaf Typha, for example. You can differentiate homogeneous areas from mixed areas. It can be really useful in sites that are especially difficult to access, traverse on foot, and it allows you to see these pockets of vegetation that you might miss with field transects. So if there's a patch of Phragmites that didn't get treated, it's easier to pick that up, and it allows for more regular sampling because it's not as dependent on your ability to access the site related to water level and that kinds of things. And because it's a UAV, you can fly under the clouds. They're not restricted by cloud-free imagery. But some of the cons, you're not going to get the level of detail in terms of species composition that you would from field sampling. Again, you can miss the understory. You're generally not going to get much information about submergent species, although we're also doing some work to get better at that. You're probably going to miss smaller, sparser, and more rare vegetation types, and you do need that access to UAV and the personnel who can operate and classify it. So overall, looking at our glyphosate Signet Plus and glyphosate Pier sites, we had similar results in terms of the reduction in Phragmites cover 1 and 2 years after treatment. 2 years isn't really a long enough monitoring period to capture our longer-term changes that we would expect in native vegetation response. We did see an immediate impact on kind of the human impacts of biomass removal, and it appears to be having a moderate positive short-term ecological impact as well. And then we found UAV mapping to be extremely useful for large and difficult to access sites, especially in terms of identifying patches of remnant Phragmites that might be missed in transects, but it is limited to the dominant vegetation, and you're going to miss understory and sparse species. So with that, I think I will turn it over to Dr. Borgeau-Chavez for more of a discussion of the satellite-based mapping.
Okay, I'm going to go into some more detail about the WorldView 2 data that we used, which is very high resolution imagery, and then a little bit more about the drone mapping. So what the remote sensing from that bird's eye view helps you determine is what that current Phragmites distribution is. Very easy to pick up in the WorldView and the UAV imagery. It helps you find where those leading edges are, and then after treatment, what the effectiveness of those treatments were. Is there standing dead that needs to be mowed? Is there regrowing Phragmites, and what are there other species that have come in post-treatment that need to be, that are secondary invaders, for example? And it basically, has the habitat been restored? So looking at the importance of scale in the imagery, on the left is the air photo from Applied Ecological Services over Saginaw Bay from 2014. And you can see the Phragmites is that kind of a grayish green color. It's purple in almost all of the classified images that I'll be showing, just like it was in what Amanda showed. And so it provides a 6-inch resolution. You can really see those leading edges. And then in the center is the WorldView, which is 2 foot. It's still doing a pretty good job of picking up where the Phragmites is. And then on the far right is a half-acre minimum mapping unit product that is basin-wide. But you can see it's at half-acre. It's really only picking up those large monocultures of Phragmites and not getting those leading edges so well. So what does the high resolution imagery allows you from that bird's eye view to delineate where exactly the frag is and where it's not. This is important for producing accurate bids for chemical or treatment or mowing or burning. Helps in locating things that should be avoided in treatment, so non-targeted wetland types. And so I'm going to go over some examples of pre and post Phragmites treatment using both WorldView and the drone. So starting with the high-res WorldView 2, which again is about 2-foot resolution. This is the overview of the classifier that we use to make all these maps. You could also hand delineate if you were able to access the image and identify the Phragmites on it. But for this, we use this machine learning classifier, random forest, and it's based on some field data that we use to interpret the image, creating these supervised polygons that then go into the classifier with the WorldView data to produce the map as shown on the right. So we did that for all of Saginaw Bay using August 2016 imagery, and that is available for anyone that's interested on our web viewer. There's a button to request download there. Also on that website, which is the main purpose for that website, is that half-acre binational wetland map that I showed in a couple of slides earlier. And if we zoom into this area, you can see this is on the map viewer, an example. On the very bottom is a button to request data. And up at the top is a button to click on just for the Saginaw Bay. So this is looking at the half-acre minimum mapping unit. And there is a difference of 6 years between these two products. You can see where Frag has expanded, but also the level of detail is much, much greater in the new product. And again, that's only for Saginaw Bay that we created that. Whereas if you go to this website, it's basin-wide with the coarser res. So I'm going to zoom into Hampton, which is located where the red box is in Saginaw Bay. And the top image and classification are before treatment. And the purples again are the Phragmites, and the mixed Phragmites is shown here as a little bit lighter purple. And then on the bottom is the 1 year after treatment where there was a lot of standing dead there. Mapped on the right, bottom right, in the gray as mixed detritus. There are 2 spots there that are showing Phragmites that was either missed or popped up after treatment. And I'm going to zoom into Finn Road, which has a pier there, and look at some ground photos. So this kind of shows what Phyllis and Amanda have been talking about, where if you're on the ground looking out, you can't really tell if there's something beyond that Phragmites. In the top, it's so dense. And then on the bottom, after treatment, you still can't really see very well at all. Standing dead, so that affected the aesthetic and recreational use of this site when that was evaluated. Then it was partially mowed in the winter of 2018, and so this bottom one shows a lot more open water and some sparse veg, and then patches of still standing dead in the grays. And so if we go back to that boat launch on Finn Road, you can see before, in the summer of 2017, the standing dead, and then after, in 2018, a huge difference that mowing made in that site, making it accessible to recreation and boat launch. So this is looking at the image, the world view image of all of Hampton. So you could just hand interpret this if you want. This shows the all of the transects that were flown with the UAV, and some of these we also did in the field. And so if you, standing from that, where that blue dot is, that is what you would be looking at, standing dead frag at that location. And then we walked out a little ways and looking back towards the pier, you can now see the pier. You can see a lot of regrowing native mixed vegetation. And this is along the shoreline there. It's mowed frag that has come into the edge, which is really kind of a hazard for animals and people too. And so then I wanted to say the use of this map. We have one example of a neighborhood on Knott Road, which decided they saw this treatment going on around them and wanted their neighborhood treated as well. And this map allowed them to determine how many acres of Phragmites they had, and allowed them to get a bid. So that's a zoom into the area. The total area was 450 acres. In the bottom, we pulled out just where the Phragmites is, and it was 235 acres that actually was treated. I don't think everyone in this neighborhood ended up having their land treated, but they all adopted the Tier 1 protocol and the photo monitoring. So these on the left are examples of some of those photos that they took. And our project ended, but they said that they would go back, and we gave them the protocols. They would go back and do the follow-up monitoring next summer. And I wanted to show this Callahan Road, which was one of our kind of trouble sites. And we're not sure if maybe because it was an old farm field, if it had maybe it had glyphosate been used when it was a farm, and maybe that's why we didn't have so much success at this site. But on the top are before treatment, and then in the bottom are post. And it's really kind of hard to see how effective it was from the photos at this site. And if you look at the world view from the 3 different years, the first one on the left, before treatment, on the yellow on the top shows the area that was being treated. You can see that this is the actual flight lines of what was treated. And if you look in the area that was missed in the second year, Phragmites popped up there. There was a little bit there that was probably missed, and then it just kind of spread. This site was treated again, and then in 2018, you can see there's a lot more frag again. So the third year, we ended up treating just this past fall with a mix of imazapyr and glyphosate, and we'll be following up with this site to see from the imagery to see if that was finally helpful in getting rid of that Phragmites there. And then just briefly, Amanda got into the drones quite a bit, but just showing these are the transects with the drone, pre-treatment drone imagery flown over, and some examples up there. You can see the Phragmites pretty clearly in that top one in the center. These transects were 850 to 1,000 meters long, and so it was really helpful to use this. We repeated the transects in October 2017 and 2018, and Amanda showed the results of that. But just looking at frag versus Typha, you can see individual Phragmites stems and inflorescences in the top image, and this is just zooming in on the top row, and then on the bottom row, it's zooming into a Typha-dominated area. This is very distinct, easy to map these 2 types in particular. A lot of the other, we just are able to map it as mixed. And then this is another site we've been monitoring, which is up at the tip of the thumb. but just due to the change in water levels at the site, the Phragmites is going away. There was no treatment here. And so we mapped where the Phragmites was, again shown in purple, and there was a change with about a 1 foot rise in water elevation between these two images. I'm sure if the water goes back down, it'll probably pop back up. So we'll be monitoring this for at least a couple more years. So just to compare the benefits and limitations of different remote sensing, different scales, the drone imagery, which again has about 2-inch resolution, is probably higher than you need, but it's a low cost. You're able to fly it underneath the clouds, so you don't have to worry about clouds being in the way, and you capture the leading edges. The same with the aerial imagery. That's probably optimum. That was applied ecological services, but that has a higher cost to have an airborne system flown. And then the WorldView 2, you have to worry about clouds, but it is free to federal agencies. We obtain these data through our Fish and Wildlife Association, an MOU with them. And that is sub-meter, and we're able to pick up many of those leading edges. And then the last one is the basin-wide map that I showed that has a half-acre minimum mapping unit. And that has a higher cost. The Landsat's free, but the Pulsar has a high cost unless you have a data grant. But that allowed us to map the entire region for understanding what the extent of Phragmites was basin-wide. So just to summarize, the aerial and satellite imagery are really effective tools in looking at that extent of Phragmites invasion. And we're able to map not only the live frag, but the dead frag, dead standing or fallen, and the leading edges if that resolution is high enough, as well as other vegetation cover types. It puts the treatment areas into that landscape context. So what is growing around it? Is there frag around the site that you're treating? Because that's propagule pressure you need to be concerned about. It's just going to seed right back in. And choosing the resolution and the data source just needs to fit the needs and the resources of the land manager. More and more often drones are available, and if you collect them in nadir, if enough overlap, you can mosaic them together and produce maps like we have done here. So you need to make sure that you choose an option that you're allowed to get repeat coverage. So we did have some issues getting WorldView imagery over some of our sites in 2017. So for some of them, we went from 2016 to 2018 without a cloud-free image in 2017. So that can be an issue with satellite imagery. But there are more and more satellites becoming available. Planet Labs, I think, collects daily or every other day. It's a little bit coarser, but that's free. And then again, I just wanted to mention again that 2016 August Saginaw Bay map is available online and for free download. And that's just our acknowledgments. Is there any questions?
Great, thanks so much, Phyllis, Amanda, and Laura. For those on the call, if you want to type in your questions into the chat box, we're a little bit over, but I think we can squeeze in a couple questions here. So the first one was from Drew, and Phyllis answered this in the chat, but I'll just share it with you all. So Drew asked if there's a difference in nutrients before and after herbicide treatments, because it seems like applying herbicide to large swaths of frag would leach nitrogen and phosphorus back into the wetlands. And Phyllis responded that they developed a lookup tool that enables you to put in nitrogen levels, water levels, and propagule pressure to determine best treatment and sequences. And then nitrogen management is essential to long-term success. We did have a webinar on the lookup tool, so you can check the recording out on our website at Great Lakes Phragmites Collaborative.net/resources/webinars, and reach out to me if you have any trouble finding that. There was another question from Glenn that WorldView is multispectral. and is approximately 1.6 meter spatial resolution. Are you pan sharpening to get 2 feet resolution? Highly technical question. Laura?
Yeah, the sub-meter is referring to pan sharpened world view imagery, which if you pan sharpen it, I think it's 0.7 meters.
Okay. Sure. Okay, thanks, Amanda. And a question from Dave. What software did you use to classify the vegetation from the satellite images? You may have mentioned that in the presentation, but what was the software?
We use Random Forest in R.
Okay, great. And then a question from Travis. What was the full link that was referenced earlier in the presentation? Maybe I can send that out to the listserv when I send out the recording, since it's going to be difficult to read out on URL, I think, on the webinar, unless there's a short code or something that you guys could share.
Oh, for which one?
The geodjango.mtri.org.
Yeah, that's a good idea to go backwards.
Yeah, we can just put that back up for a minute.
Okay, perfect.
And then Phyllis had on her slides our website too. Oops.
Okay, there it is.
She has the report on it, but her slides seemed out of sync, at least for us. So I don't know if other people had that problem too.
Okay, yeah, there might have been a delay with the software, but the URL is up there now on the left side of the screen. If there's any more questions, type them into the chat box now. And I just have one quick question. We often get requests for people who are new to managing Frag and are looking for a simple monitoring tool. Would the photo monitoring seem kind of accessible to those who are new to monitoring? Is there a 1-pager or a 2-pager, some sort of brief summary of that technique that we could share out with the listserv, just as a getting started option for monitoring?
We could probably turn it into one. We have a appendix of our report that shows all the monitoring tiers.
Yeah, kind of the step-by-step protocols for all of these.
So I could share that.
Yeah, that would be perfect. And if you can share that with me, and then I'll send it out with the recording.
One thing that I would suggest is that we also develop a little handout that shows all of the invasives to be concerned at, because if they can learn those, then they can do early detection for those as their treatment progresses. And we have not developed that yet.
Okay. And for those looking for more information about the specifics with the remote sensing monitoring, is there a website or a report that would be the best resource for people to look to start with?
So for someone to do it themselves, is that what you're thinking?
Yeah, and maybe to plan it themselves and maybe have a contractor do it or something like that, but just something to kind of get started for those who are new to the method.
Yeah, I think we included that too in the protocols with like how high we flew the UAV and what the overlap needed to be. I'm pretty sure that I'll double check that it's in there. If not, I'll find it and get that to you.
Okay, perfect. All right, so it looks like that's all of our questions. Thanks so much to Amanda, Phyllis, and Laura for your presentation. I will upload this recording for those who missed it or who would like to watch it again to get to see some of the details again. Thanks to those who joined us on the call, on the webinar. We do have another webinar coming up, I believe in August, Laura, that's part of this project as well. Do you want to just speak about what that will be about?
Is that Collins? I'm trying to think what it is.
Yeah, I believe so. And yeah, I could be mistaken, but I thought it was sort of associated with this work in some way.
Yeah, I didn't know if I think he did set one up. So it's broader than this. So we had a project with the USGS and PAMP, and he collected UAV over the PAMP site. So I think that's what he's presenting on. And so that would kind of go over the step-by-steps of how high to fly and what the overlap needs to be and what software we use to mosaic the images and produce a DEM and stuff like that.
Okay, that's great. Yeah, and that'll be a good resource too for people getting started to have some more of those details. Okay, thank you. So those on the call, I will be sending out the information about that webinar as we get closer. And those who are listening to the recording, you can probably check it out soon on our webinar archive or on our YouTube channel as well. So thanks again to our presenters. Really appreciate you sharing your work with us, and lots of optimism from Phyllis. I appreciate you starting the presentation by reminding us all there's a light at the end of the tunnel with Frag, and by monitoring to make sure we're being effective, we can move towards getting those native systems back. So thanks so much for that, and we'll conclude today and hope everyone has a great day.
Thank you.
Thanks.
Thanks everybody. Bye-bye.